Friends, welcome back to my channel Only Bio. Continuing with the series of the NEET 2020 paper, the MCQs being solved from question number 71 to 80 with detailed explanation. The first question relates with the chapter Morphology of Flowering Plants, and the question is: the ovary is half inferior in dash. A. Mustard, B. Sunflower, C. Plum, D. Brinjal. And the right answer for this question is C. Plum. Look on the screen to understand what is meant by half inferior ovary, which is also called as the half superior or sub inferior or partially inferior ovary, and the flower is said to be perigynous. Now, in this, a half inferior ovary, we can see that it has nearly equal proportions of ovary above and below the insertion point. Other varying degrees of inferiority can be described by other fractions. Only 1/4 portion of a 3/4 inferior ovary is above the insertion. This type of organization is found in the flowers of plants such as peach, plum, rose. Now the option mustard is the one which has hypogynous flower. Epigynous flowers are seen in case of array florets of sunflowers. So even that can be the answer. Okay. So this is the reason why the answer is plum. This question relates with the chapter Biochemistry of Cell. Identify the basic amino acids from the following: A. Glutamic acid, B. Lysine, C. Valine, D. Tyrosine. And the correct answer for this question is B. Lysine. Friends, remember each amino acid has an amine group at one end and an acid group at the other end, and a distinctive side chain which is called as the R chain, that is variable. The backbone is the same for all amino acids, while the side chain differs from one amino acid to the next. Okay, so depending on this, we have amino acids which can be categorized as acidic, basic, or neutral. Now, amino acids which are typically acidic are glutamic acid and aspartic acid. The one which are basic include arginine, which is strongly basic, histidine, and lysine. And rest all are the examples of the neutral amino acids. This question relates with the chapter Circulation. Match the following and select the correct option. We have two columns. Column 1 having A as eosinophils, B as basophils, C as neutrophils, and D as lymphocytes. Column 2 having 1 immune response, 2nd phagocytosis, 3rd release histaminase destructive enzymes, 4th release granules containing histamine. And these are your options. The correct answer for this question is the fourth one, that is eosinophils release histaminase, that is the destructive enzymes. Basophils release granules contain histamine. Neutrophils are involved in phagocytosis, and lymphocytes are involved in the immune response. Friends, eosinophils, which we also call it as acidophils, we know they're a part of, or they're a type of WBCs, and they're a part of immune system, right? Along with the mast cells and basophils, they control mechanisms associated with allergy and asthma. These are important mediators in allergic responses, you can say. Now, basophils are also a type of WBC. They're the least common type of granulocyte you might be recollecting, which are responsible for inflammatory reactions during immune response, as well as in formation of acute and chronic allergy diseases, including anaphylaxis, asthma, atopic dermatitis, and hay fever. They coordinate the immune responses, including histamine and serotonin, that induce inflammation. Then the next one is neutrophils. which are also at times called as the heterophils or neutrocytes. They also form an important part of innate immune system. Okay, they're phagocytic. That is, during the beginning or acute phase of inflammation because of any bacterial infection, neutrophils are the first responders of inflammatory cells to migrate towards the site of inflammation and bring about phagocytosis. And the option D, that is lymphocyte, it is also subtype of WBC, which includes natural killer cells, that is cytotoxic cells, yeah, T cells and B cells, which are involved in the immune system. Okay, so there are two main types of lymphocytes, you can say the T cells and the B cells that produce antibodies as well as attack the bacteria, viruses, and toxins. This question now relates with the chapter. Actually, we can't relate it directly with the chapter, but थोड़ा बहुत इसको biochemistry से हम relate कर सकते हैं, or we can categorize it as an additional question. It includes match the following. Column 1A, inhibitor of catalytic activity. B, possess peptide bonds. C, cell wall material in fungi. And D, secondary metabolite. In column 2, at the first position is ricin, second malonate, third chitin, and fourth collagen. These are the options given here, and the correct option for this question is the fourth one. That is the first A, inhibitor of catalytic activity is malonate, possess peptide bonds is collagen, cell wall material in fungi is chitin, and secondary metabolite is ricin. Friends, ricin, it is one of the most poisonous substance on the earth. It's dangerous in about the way it gets in your system by ingesting it. Okay, it is about the least dangerous way when it is ingested, you may say. Injecting or inhaling requires about 1,000 times less ricin to kill a human than ingesting it, and that's a very small amount. Perhaps you might have heard about it recently. Ricin was laced up on some envelopes which were being addressed to one of the American president. I hope you got the hint, right? The seeds, I mean, ricin ye kahan se milta hai? It is obtained from the seeds of the castor plant. Yes, the same from which we get the castor oil, but the castor oil doesn't have any problem. Ricin is a toxic protein which infects the cells and blocks its ability to synthesize its own protein. And inhalation has a different impact, okay, and ingestion has a different impact. Please remember the castor oil wouldn't have this kind of a problem. However, ricin is this toxic protein that it is also used as a bio weapon. Okay. Now the second option here in column 2, that is malonate. It is reversible inhibitor of succinate dehydrogenase enzyme, which plays a central role in the TCA, that is Krebs cycle, and is a part of complex 2 of the electron transport chain. Malonate compounds include salts and esters of malonic acid. Now, the option 3 from column 2 is chitin, which is a fibrous substance consisting of polysaccharides, the major constituent in the exoskeleton of arthropods and the cell walls of fungi. And collagen, the option 4th from column 2, is the most abundant protein in the human body that is found in the bones, muscles, skin, and tendons, and it is the substance that holds the body together. Okay, it provides strength and structure. Now, this question relates with the chapter cell division, and the question is identify the correct statement with regard to G1 phase of the interphase. G1, that is gap 1. A, reorganization of all cell components. B, cell is metabolically active, grows but does not replicate its DNA. C, nuclear division takes place. And D, DNA synthesis or replication takes place. And the correct answer is B, that the cell is metabolically active, it grows but does not replicate its DNA. Now, G1 is an intermediate phase, it occupies the time between the end of cell division in mitosis and the beginning of DNA replication during S phase. So during this time, the cell grows in preparation for DNA replication, but it does not replicate its DNA. It may also increase some intracellular components such as the centrosomes that undergo replication. Now, this question relates with the chapter plant growth, and the question is name the plant growth regulator which, upon spraying on sugarcane crop, increases the length of the stem, thus increasing the yield of the sugarcane crop. Option A gibberellin, B ethylene, C abscisic acid, and D cytokinin. And the correct answer for this question is A, that is gibberellin. In successive years of planting sugarcane, the internodes they start shortening, and this may result in production loss. Okay, now this process is difficult to reverse, and the use of plant growth regulators only can solve the problem. Now, for promoting the sugarcane growth, gibberellins can be used up because they adjust the function of sugarcane, improving the carbon oxidation of the plant. So, according to one experiment done on sugarcane, injection of gibberellin in the sugarcane had increased the length by 5 to 10 centimeters. Okay, that's quite a big number, you can say. So, the sugar content was increased by approximately 5%. And this is the reason why gibberellin technology is widely used. Now, this question relates with the chapter genetic basis of inheritance. The question is identify the wrong statement with reference to the gene I that controls ABO blood groups. A person will have only two of the three alleles. B, when IA and IB are present together, they express the same type of sugar. C, allele I does not produce any sugar. And D, the gene I has three alleles. And the correct answer for this question is B. Friends, remember in humans, the ABO blood groups are controlled by a gene called as gene I that stands for isoagglutinin. And this I has three alleles, namely IA, IB, and small I. So a person possesses only two of the three alleles. Now IA and IB dominate over small I. But when they are present with each other, they act as co-dominant genes. And these alleles help to determine the blood group of the person. मतलब IA, IB, small I, ये कौन से combination में आएगा? This will help us to understand our blood group. Now these alleles, they help to determine the blood group, but how? The reason is the plasma membrane of the red blood cells has sugar polymers which project out from its surface, and the kind of sugar produced here that projects out is regulated by the gene I of the ABO blood group. So the alleles IA produces A type of sugar, the allele IB produces B type of sugar, while the allele small I does not produce any sugar. Just I also is never going to produce any. Now this question relates with the chapter Human Health and Diseases. Identify the wrong statement with reference to immunity. A. When ready-made antibodies are directly given, it is called passive immunity. B. Active immunity is quick and gives full response. C. Fetus receives some antibodies from mother and it is an example for passive immunity. D. When exposed to antigen, living or dead, antibodies are produced in the host's body and it is called active immunity. And the answer for this question is B. That is, active immunity is quick and gives full response is a wrong statement. Now let us understand it. See, the option A, when ready-made antibodies are directly given to protect the body against foreign agents, it is called passive immunity. That's a correct statement. Option B, active immunity. Please remember it is slow and it takes time to give its full effective response. Injecting the microbes deliberately during immunization or infectious organisms gaining access into the body during natural infection definitely induces active immunity. Now the option C, mother's milk is considered very essential for the newborn. The yellowish fluid colostrum secreted by the mother during the initial days of lactation has abundant antibodies called as the IgA to protect the infant. And the fetus also receives some antibodies from the mother through the placenta during pregnancy. And these are the examples of passive immunity. Now, option D, when a host is exposed to antigens, matlab any substance which the body recognizes as foreign या alien and induces an immune response, which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. And this type of immunity is called as active immunity. Now, this question relates with the chapter Human Nutrition. The question is, the enzyme enterokinase helps in conversion of dash. A. Trypsinogen into trypsin. B. Casinogen into casein. C. Pepsinogen into pepsin. And D. Protein into polypeptides. And the correct answer for this question is A. Trypsinogen into trypsin. Now, friends, enterokinase is an enzyme which is also called as enteropeptidase. This is produced by the cells of the duodenum and is involved in digestion in humans and other animals as well. Now, enterokinase, it converts trypsinogen into its active form called as trypsin, and this results in the subsequent activation of pancreatic digestive enzymes. The absence of this enzyme results in intestinal digestion impairment. Now, this enzyme, that is enterokinase, was discovered by Ivan Pavlov, and it is the first known enzyme to activate other enzymes, and it remains a remarkable example of how serine proteases have been crafted to regulate the metabolic pathways. Now, this question relates with the chapter genes, and the question is, the specific palindromic sequence which is recognized by EcoR1 is dash. We can also relate this question with biotechnology. Option A, you'll have to read this out. B, C, and D. The correct answer for this question is D. That is, at 5-N we have GAATTC3-. From 3 to 5 we have CTTAAG5-. Now, EcoR1, is a restriction endonuclease that is isolated from Escherichia coli. The eco part of the enzyme's name originates from but obvious Escherichia, and the R represents a particular strain. That is, in this case, it is RY13 strain. The last part of its name, that is I, denotes that it was the first enzyme. Okay, EcoR1. Okay, actually, it is the first enzyme isolated from the strain. And this is a restriction enzyme that cleaves the DNA double helicases into fragments at specific sites. So, EcoR1 creates 4 nucleotide sticky ends with 5-N overhangs to of AATT, and remember that it generates sticky ends of DNA which are often used to cut the DNA prior to ligation, as the sticky ends make the ligation reaction efficient. Friends, like, subscribe and hit the bell icon if you haven't done it. Thank you so much.